It's a great pleasure to be here. Thank you very much for inviting me. It's slightly strange not being at a Gordon conference with you all and instead being in my attic, but that's where we are at the moment. So I'm going to be talking about sphingolipids from the perspective of diseases and how diseases can give us some insights into the complex roles of sphingolipids. So as we're all here because we've got a shared interest in these molecules, and it was through the lysosomal disease field that I became interested in sphingolipids. And I think that, you know, we've heard over the last few presentations in this excellent series that we've made a lot of progress in being able to measure these quite challenging lipids. We've understood a lot about their biosynthesis and the enzymology of how they're made. And we've got a fairly detailed understanding of their catabolism in the lysosome. But one of the things that Tony alluded to in his talk is that as we've worked on sphingolipids over the years as a field, the complexity and the sophisticated fine-tuning of this system that we don't fully understand has really been a major challenge. And I think Tony in his talk was sort of subliminally implicating divine intervention in some of this complexity. And Tony knows me well enough that I can put myself in this other schematic with, you know, there's clearly been considerable evolutionary pressure driving this diversity of sphingolipids. And the real challenge is, how do we understand more about sphingolipid function? And I'm not professing to have done that in my career in terms of the fine detail of how sphingolipids work. But I think studying diseases where imbalances in sphingolipids occur is giving us some clues. And also, I don't know if Tim Cox is on the call, but Tim is, I remember giving a couple of talks entitled Treasure or Exceptions. And this is a book about the life of William Bateson, who advocated the power of studying inborn errors of metabolism to try and understand fundamental processes. And so a lot of our work has been to study rare genetic diseases and try and use that information to do two things, really. One is to understand more about sphingolipid functions by understanding what happens when things go wrong. And the second is to use that knowledge to try and innovate new therapies. So the diseases that I'm going to be focusing on for almost all of the talk, but not the end, are single gene disorders. And in fact, most of these sphingolipid disorders, both the biosynthetic ones and the catabolic ones, are typically inherited as autosomal recessive traits. And they're called simple genetic diseases because there's one gene, and the gene usually encodes something that we understand the function of it. But I think, as you'll see in my talk, there's very little simple about these disorders. And I think this is a bit of a wake-up call for the challenge of trying to understand polygenic diseases where there may be, you know, multiple, you know, gene variants and environmental influences on disease presentation. So I'm going to begin by just focusing on single gene disorders. Now, Rick Proyer published many years ago a really sort of landmark paper that showed that if you knock out glucosylceramide synthase, it's embryonically lethal. And this really re-emphasized the fact that glycosphingolipids are needed for a variety of functions. And this embryonic lethality led Rick to say in this paper that the glycosphingolipids are involved in crucial cell interactions mediating these developmental processes that failed in terms of embryonic development in this mouse model. Now, I think one of the things that this paper did, apart from emphasizing the importance of glycosphingolipids, was it raised a really interesting question as to whether or not it was any perturbation in glycolipid biosynthesis would be embryonically lethal, or whether it was only when you knocked out the whole pathway. And the story in terms of the biosynthetic disorders that we've worked on is an interesting one, because if you look at glycolipid biosynthesis, and here's the enzyme that Rick had knocked out, and this was embryonically lethal, I think many people in the field imagined that this was probably telling us that any enzymatic deficiency in this pathway could also be embryonically lethal, so wouldn't present as a biosynthetic disease. But I remember having a call from Andrew Crosby. He's a geneticist. He worked at St George's and is now at Exeter University. And Andrew was mapping a very severe epilepsy disease in an old Amish population in Ohio in the USA. And the reason that Andrew contacted me was he was slightly panicked because he had mapped the gene for this disease and then gone to the literature and thought that he then doubted whether the gene could be correct, because his view was that there could be no biosynthetic human diseases. And in fact, Andrew was absolutely right. The gene in question was responsible for a disease. And he actually went on to map two diseases. One is Gm3 Synthase Deficiency, which is in this blue box. You lose these complex gangliosides. And then went on to find another disease, which is a spastic paraplegia, which is a Gm2 synthase deficiency that has this broader range of defects in terms of sphingolipid biosynthesis. So what this says is that the whole pathway is needed for embryonic development and life, but you can have defects in these downstream enzymatic steps in biosynthesis that lead to neurological diseases. But as you can see, very, very different diseases between this severe epilepsy and spastic paraplegia. And Andrew Crosby's phone call triggered us to, through David Priestman's work in the lab, to go on to show biochemically that his gene assignments were correct and to prove that this really was Gm3 and Gm2 synthase deficiency. And this was published in a couple of papers several years ago. So those of you who are new to the field, this is just to alert you to the fact that we have got these two interesting diseases that have arisen through studying these very unique human populations. However, those studies and those diseases really lead to a number of, at the moment, unanswered questions. And that is that what's the mechanism through which those enzyme deficiencies lead to such severe human disease? I mean, it's very tempting to think that it's the loss of the complex gangliosides and their functions. But equally, what happens in these disorders that David discovered with his biochemical work is that the absolute amount of glycosphingolipids remains fairly constant, but you've now got an accumulation of precursors. So the balance between the complex gangliosides and the precursor glycolipids is greatly imbalanced in these disorders. So are the precursors responsible for the biological deficits in this disease, in these diseases? Also, we don't know anything about the distribution of these lipids and their localized levels within the cell. And also, Rick Preuer, at the time that we published the Gm3 synthase paper, wrote a News and Views article where he raised a very interesting possibility about whether it's a change in the neutral to charge balance in neuronal membranes that is really perturbed in these disorders. And obviously, a knowledge of these mechanisms is really important if we're going to develop routes for therapy in the future. So this is really just to alert you to the fact that these biosynthetic diseases occur. They're extremely interesting, and there are a lot, if you're new to the field, there's a lot of space for doing some very innovative work on mechanism in these disorders. Now, most of my career I've spent on sphingolipid catabolic diseases, and these are the lysosomal storage diseases. So we're all familiar with the concept that the sort of reverse of the biosynthetic pathway happens in the lysosome, where you degrade glycosphingolipids and sphingolipids in general. And we've got a very good understanding of the enzymology of this sequential degradative pathway. The enzymes are in red and the diseases are in blue. And this is just an example of a Tay-Sachs electron micrograph showing you these spontaneous formation of membrane inclusions in lysosomes due to the accumulation of Gm2. So this set of disorders are much better understood and well characterized than the biosynthetic diseases. And most of them are autosomal recessive, although there are a few that are X-linked. There's probably well over 70 that are now known, and there's an interesting group of diseases that are secondary lysosomal diseases that have a different mutation. So the mutation is not in a lysosomal enzyme, for example. They're individually rare, but they collectively occur at about 1 in 5,000 live births. About 3/4 of them are the result of defects in lysosomal enzymes. But we've had a particular interest over the years in lysosomal membrane protein defects, because they also lead to disease. And much like other neurodegenerative diseases, they have neuroinflammation as a major component. And again, about 2/3 of these diseases involve pathology in the central nervous system. So the functioning of our brain, spinal cord, peripheral nervous system is really very dependent on the correct homeostasis of this class of lipids. Some of the lysosomal storage diseases, although they're rare, have approved therapies. Some of those are now generics, and there's a lot of activity in this field, developing new innovative therapies, and I'm sure many of you are actually involved in that who are on this seminar. Now, another thing to bear in mind is that when you have a defect in the lysosome, it affects the lysosome in all tissues, and so it's not surprising that the lysosomal storage diseases are multimorbidity diseases, and that is that the disease is present in multiple organs and tissues. And this is a schematic from a review. It's a schematic that Cynthia Tift made, and just to illustrate that if we look at these boxes, you can see that the brain is the largest box. Each of these italicized series of letters is a gene, and you can see that the brain is affected in most of these disorders, but so are a number of other tissues like the lung, the hematopoietic system, joints, the skeleton, and the visual system. And so if we just look at Niemann-Pick C, which is a disease that we've done a lot of work on, it affects the central nervous system, it affects the lung, it affects the visual system, and it also affects the hematopoietic system. And you could actually add probably 3 or 4 circles to most of the genes that are listed here. So these are complex disorders, and from a clinical point of view, although the brain may be the primary organ that's affected, we really need treatments that affect all of these compartments if we're going to make a difference to patients long term. So I'm going to focus on Niemann-Pick C, which is a disease that we've worked on a lot over the last sort of 10 years. And it's a very enigmatic disease that it never ceases to amaze me how complex it is. So it's, at one level, it's very similar to most lysosomal storage diseases in that it has, it affects about 1 in 100,000 live births. The cerebellum is affected, you get Purkinje cell loss, you get neuroinflammation, and patients develop ataxia and dementia. And interestingly, in areas where most of the lipid storage is, they get pathology that looks like sporadic adult onset Alzheimer's. So there's something about the cell biology of this disease that accelerates tau and APP processing changes. Now, where this disease differs from other lysosomal storage diseases is that it can be caused by mutations in 2 independent genes, NPC1 and NPC2, and also the spectrum of lipids that are stored in this disease are very broad, and cholesterol is one of them, as are all of the sphingolipids. So this is a complex lipid storage disease, and it has rather unusual genetic basis. One of the main features of the reason for the cholesterol accumulation is that when LDL-derived cholesterol is taken up into cells, normally that would be delivered to the ER, and that happens very, very inefficiently in this disease. So there's a sort of a block in cholesterol delivery to the endoplasmic reticulum. And also the sphingolipids also build up in sort of late endosomes, and there's a block in late endosome lysosome fusion that's quite unusual and is a feature of this disease. And at the moment, there's only one approved therapy, which is a substrate reduction therapy drug, miglustat. It's a glycolypic biosynthesis inhibitor. And it's not totally clear why this drug works in this disease. But there are other treatments that are both in clinical trials and also that have completed clinical trials recently. So we're hopeful that the treatment of patients will improve over the next few years. Now, if you look at these proteins, this is a schematic where we're not looking at a cell here, we're just looking at the lysosome. This is the limiting membrane. And if you look at the protein products of the NPC1 and NPC2 genes, NPC1 sits in the limiting membrane of the lysosome, complicated multi-membrane pass protein with these luminal loops. And NPC2, on the other hand, is a housekeeping protein. It's mannose-6-phosphorylated, and it's called combined cholesterol. And there's also a cholesterol binding site on NPC1. Now, NPC2 was actually, when these two genes were cloned, this was a novel finding, but actually NPC2 had been known for a number of years as HE1, a protein that's found at very high levels in the human male reproductive tract. It's called human epididymal factor 1, and it's about 20% of the the proteome in the testis is actually NPC2. So it's a very, very high, highly expressed, and it's thought to be the donor of the cholesterol that's needed for sperm maturation. What we hadn't appreciated until this disease was identified is that it's actually ubiquitous in all of our lysosomes, and the relationship between these two proteins still remains a matter of research to date. Now, one of the really fascinating things about NPC1 is a few years ago it was identified that it acts as the Ebola receptor. So Ebola virus it comes into cells almost passively through a rather generic interaction of the plasma membrane. It's endocytosed, and the second luminal loop of NPC1 acts as its receptor. And it was actually the first intracellular viral receptor to be identified. And what this Ebola needs is an intact NPC1 second luminal loop. And so if you look at patient cells or you look at mice that are mutated in this, they're all resistant to Ebola. So this is a question as to, evolutionarily speaking, why has Ebola decided to use this as its receptor? And one of the links is that Ebola also needs calcium release from the lysosome in order to complete its life cycle. And there are now links between NPC1 and calcium transporters in the lysosome that have come out of some yeast screens that we've been doing. So it's possible that this is, actually confers a couple of advantages to Ebola to use this as its receptor. Now, NPC1 continues to be an interesting hub for infectious diseases, because we found a few years ago that Mycobacterium tuberculosis sheds lipids that inhibit this pathway. And by doing so, by inactivating this protein, it blocks phagosome lysosome fusion. So this is a really interesting new avenue of research in our lab. And we've just recently identified the fact that lipids shed by Mycobacterium tuberculosis, all strains that we've studied to date have the ability to inhibit this protein, and we're trying to understand the precise inhibitor structure in collaboration with colleagues at Imperial College. Now, if you go to the reviews on the subject of the MPC pathway, you'll often see schematics like this. So this is just showing that LDL, this is the lysosome again. If you have LDL-derived cholesterol, it gets cleaved to free cholesterol, binds to the NPC2 binding site for cholesterol, and is transferred to NPC1, and then in some way gets out of the lysosome and then moves to the ER. And obviously there's going to be some sort of protein required to shuttle this to the ER. And so far, the cytosolic cholesterol binding protein hasn't been identified. So there's a slight question mark over this, this schematic. A number of years ago, we found that this was quite complicated in the sense that if you look at NPC1 and you inactivate it, you see a very rapid increase in sphingosine levels, suggesting that this may be a sphingosine transporter. And this then leads to a drop in calcium levels in the lysosome. So the lysosome is the third regulated calcium store in the cell after the ER and mitochondria. And it's this calcium release that seems to act locally to facilitate fusion events and trafficking events. And it's this calcium release from the lysosome that Ebola also needs to complete its life cycle. So NPC is characterized by about 30% of the wild type levels of calcium in the lumen of the lysosome. And it's this failure to release enough calcium that then several hours later blocks late endosome lysosome fusion and leads to cholesterol and glycolipid storage. So I think that this pathway and the relationship between NPC1 and NPC2 is quite complex. And it's possible that this is a cholesterol regulated system rather than being a, if you like, a bona fide cholesterol transporter. Now, more evidence for the role of sphingosine in this disease was provided by Doris Hoglinger. Doris was in a Sphingonet network with many of you, I'm sure, who are on the line. And Doris developed a beautiful chemical probe in Carsten and Schultz's lab. And she also worked as a fellow in Oxford before setting up her own lab in Heidelberg. And what Doris did was she made a trifunctional sphingosine probe, and she was able to uncage it and look at the signal in control cells. And you can see that at the first point that you can measure the signal, you've got the start of a punctate lysosomal distribution. And by 10 minutes, you've got this beautiful ER filamentous network lit up with this probe. When Doris treated wild type cells with an inhibitor of NPC1 or silenced NPC1, you can see that the signal remained punctate. In other words, the sphingosine was never leaving the lysosome. And she took this study one step further and looked at a panel of fibroblasts from different Niemann-Pick C patients. Here's the control, which looks like this. And now you can see a mild patient. It looks a bit more punctate, but does move out to the ER. But in these moderate and severe patients, a lot of the signal remains punctate. So we think that there's a slowing in the efficiency of sphingosine export from Niemann-Pick C cells. Now, quite recently, the yeast ortholog of NPC1 was crystallized. This protein is called NCR1. There have been some mammalian NPC1 structures elucidated, but the NCR1 has given us the major insight into how this protein works. And what is proposed is that NPC2 binding cholesterol in the lumen of the lysosome transfers it to NPC1, and there is this tunnel-like structure that can accommodate cholesterol, possibly also sphingolipids, and move them into the inner leaflet of the limiting membrane of the lysosome. And so this is just shown as a cartoon over here. Now you'll notice that this is moving cholesterol from a luminal site into the limiting membrane, but it is not effluxing it in the traditional sense. So this crystal structure actually supports a model whereby this is actually moving cholesterol into the limiting membrane rather than acting as an effluxer. So this begs the question of what's going wrong in Niemann-Pick C disease. And we know from the lipid field that the most efficient bulk movement of lipids happens at membrane contact sites. And there are a whole variety of lipid transfer proteins that have been identified between different organelles. And so one of the things that we wondered was, if we've got in Niemann-Pick C a failure of transport of cholesterol and sphingolipids from the lysosome to the ER, one very simple explanation is that in some way NPC1 is important for forming contact sites with the ER. And if there's a failure in those contact sites, that would explain why there's a failure in movement of cholesterol to the ER, whereas even in NPC deficient cells, you can get cholesterol delivered from the lysosome to, for example, mitochondria. So this was our hypothesis. And essentially what we were saying is Niemann-Pick C a disorder of a failure in lysosome ER membrane contact sites? And I think, again, for those of you new to the field, this issue about how organelles talk to each other, how they move lipid substrates between them, I think is the next major frontier in this field. I think it's a really, really fascinating area of research, and we're only in the infancy of understanding this at the moment. And this again was work by Doris when she was a fellow in the lab, and has been also driven in a major way by Emily Eden at UCL, who's a collaborator. Now, the the sort of insight that we may be on the right track in terms of the contact sites came from the work of Ali Calesso, who is also in the same Sphingonet network as Doris, a phenomenally productive network. Maya Hoglinger at the Weizmann Institute was one of our partners, and we had a wonderful exchange where Ali, who was a PhD student in my lab, went to Maya's lab and ran some screens studying the ortholog of NPC1, this NCR1 protein. So this is the protein that the crystal structure has been done on. And what she did was she did a split reporter assay in yeast using NCR1 as the bait. And what she looked at was for direct interacting partners, proteins that could be anywhere in the cell. And what Ali found was that she identified a lipid transfer protein anchored at membrane contact sites 4, this LAM4 protein, as a strong interacting protein. And these LAMs are ER lipid transporters, and they've got a couple of motifs, a gram domain and a start-like domain. And if you look in mammalian systems, the equivalent proteins are these gram D1A, B, and C proteins, which contain the same domains. So this suggested that potentially NPC1 may interact with one of these gram D1 proteins in mammalian cells. And the advice Maya always gave us whenever we did anything in yeast with her, was she'd say, go and verify it immediately in a mammalian cell. Don't do anything more in yeast, go to the mammalian system. And that's what we did. And so what Emily did was that she first of all looked to see whether or not you could pull down gram D1 proteins as interacting partners with NPC1, and the signal that she got was that it was gram D1B that was the interacting partner in human cells. Very interestingly, this interaction was greatly reduced if you grew the cells in lipid depleted media. So it was suggesting that this is a lipid stabilized or promoted interaction. And what Emily also did was that she silenced NPC1 and she independently silenced gram D1B, and you can see that they phenocopy each other in terms of reducing cholesterol ester formation. In other words, they were showing the same phenotype, whether you were inactivated gram D1B or you silenced the NPC1. So this was suggestive that this may indeed be relevant to understanding NPC disease. So what Emily did was looked at ER lysosome contact sites using quantitative EM, and she found that the gold that's picking up NPC1 was overrepresented at membrane contact sites versus non-membrane contact sites. And so this suggested that you were, if you like, accumulating NPC1 at the sites where you have this juxtaposition of ER and lysosomes. Now, one of the things that really, really amazed me was when I was talking to Emily about these data was, I asked her the question of, well, how many lysosomes do you find in contact with the ER at any given time? And she said it's about 80%. So this is a very, very normal interaction. These are transient interactions. And, you know, I think one of the things we're doing when we're looking at subcellular fractionation and things is that we're probably seeing this as a contaminated preparation. And we may actually be underanalyzing these kind of contact site where two organelles are talking to each other in some of the biochemical work that we do. And this was a bit of a wake-up call to me. So she looked to see whether or not contact sites were reduced in the NPC fibroblasts, and that was indeed the case, which fitted with the rest of the data. And another thing that came out of this study that I think is, again, quite extraordinary was that Emily started seeing at the EM level these kind of structures. So this is a mitochondria. And if you look within it, you can see what we would all look at and say is a lysosome. And here's another one. You can see this sort of this engulfment of the lysosome by mitochondria. And this is just showing you on this video that you can see that this is interconnected. It's that the mitochondrial membrane is wrapped around this. It's not engulfed in a strict sense, but you can see in the Z-stack reconstructions that these are connected to the cytoplasm. But what this is suggesting is that when you don't make the right balance of lysosome ER membrane contact sites, you start making inappropriately long-term contact sites with mitochondria. And we know that mitochondria are getting too much cholesterol and are certainly dysfunctional in a number of ways in Niemann-Pick C disease, and this gives us some insight into why that might be the case. So what the model that we're now working on is that the NPC1 protein and the GRAM-D1B form a tethering complex, and Doris and Emily went on to show that if you artificially tether with other protein pairs, you can actually get a normalization of Niemann-Pick C cells using that approach. So our hypothesis is that the movement of lipids out of the lysosome is happening at these contact sites, and Niemann-Pick C1 is one of the players in this. And at the moment, Emily's working to determine which of the, where the binding site is for the GRAM-D1B protein. So in addition to the yeast models and studying patient cells, one of the other ways that we've gone over the years in trying to understand sphingolipid functions better is, and to try and understand pathogenesis, is to study what happens with therapy. And this is a much more cyclical process than you might imagine, because when you treat somebody and you discover that certain things respond and other things don't, it gives you insights into the role of these lipids in different tissues. And also, sometimes there can be clinical observations made that actually reveal fundamental insights into basic science. So this is a very much a dynamic interaction, and one that we've used in a number of ways over the years. And I'm just going to share with you one example of some work that's been really, really fun to unravel. So if you look at the therapeutic pipeline in Niemann-Pick C, miglustat in blue is the only approved drug so far, but there are HSP70 upregulators like arimoclomol that have just completed phase 3 trials and are under consideration by the FDA at the moment. Ahad Rahim has shown that gene therapy is beneficial in NPC mice. There are a number of other therapies, including cyclodextrin therapy, that have been used to reduce cholesterol burden in NPC, and these have also are in clinical trials. And also things like anti-inflammatories and things work. So this is quite a rich sort of tapestry of developments in the NPC therapeutic space. But where we've placed these different arrows is slightly arbitrary, because we still don't really understand even how miglustat works in this disease. And it had always been thought that by reducing glycolipid levels, that this was the basis for the improvement. But in fact, miglustat, which we know is a glucoside synthase inhibitor, and it crosses into the brain, it reduces complex glycolipids, and we certainly see that particularly in peripheral tissues in NPC. But very interestingly, Hans Ertz and Seng Cheng and colleagues showed that paradoxically, GBA2 might be the target. Miglustat is a GBA2 inhibitor, a very potent inhibitor, and there's evidence in the mouse model that this may be the target. So if you think about this therapeutic pipeline, even though you've got an approved drug that we know works, we don't necessarily know exactly how it's working. And that's something that's actually quite complex to unpick in these diseases. So the link from the clinic to shed further light on these rare sphingolipid disorders came from a misdiagnosis of a patient. And miglustat is the first-line therapy for treating Niemann-Pick C patients, and a lady in Italy, an adult with a quite complex disorder, presented and was misdiagnosed as having Niemann-Pick C, and she was put onto miglustat treatment. And unlike when you treat a patient, a Niemann-Pick C patient with miglustat, you slow disease progression, but you don't completely arrest or reverse pathology. However, this patient responded in a quite spectacular way, in the sense that she had peripheral neuropathy, she had skin lesions, she had edema in multiple tissues, and those cleared up on treatment. And this patient came to the attention of Bruno Bembi in Trieste, and Bruno knew immediately that this was not Niemann-Pick C, because he's an expert in lysosomal disorders. And he and Andrea Dardis worked out that this lady actually had Tangier disease. And Tangier disease is an ultra-rare disease. It's named after Tangier Island in Chesapeake Bay in the US. And what we were wrestling with was, why does miglustat treatment correct a completely unrelated disease from Niemann-Pick C? And the clinical data suggested that perhaps NPC and Tangier cells may have convergent pathogenesis at a certain level. And so this work, I think I've just been invaded by a wasp or something here. This work was done by Ali Kaleso again and H.M. Kaya, who they were two fantastic PhD students in my lab who are now postdocs. And so they set about to have a look at the pathogenic cascade in NPC and Tangier disease cells to try and understand what this therapeutic benefit was coming from. Now, ABCA1 is the gene that's responsible for Tangier disease. It's obviously well known in its reverse cholesterol transport role in things like macrophages. And in these patients, the classical infantile onset patients from Chesapeake Bay, Tangier Island, have point mutations, whereas some of these adult onset forms of the disease have a large deletion, yet they have a later onset presentation. And what Ali and HM did was they looked to see biochemically whether there were similarities between Niemann-Pick C cells. And you can see that sphingosine storage is a salient hallmark of NPC, and we saw it in 3 Tangier disease patients. Also, there's a more modest elevation of sphingonine. The thing that's quite unique about Niemann-Pick C is that it has this attenuated calcium store homeostasis. I said that the lysosome only has about 30% of the calcium content as wild-type cells in Niemann-Pick C. And what was very interesting is that these 2 patients who have ABCA1 mutations have exactly the same calcium defect. So you release selectively from the lysosome. Here's the wild-type curve. Here's in purple is the NPC showing this attenuated release. And you can see that this is superimposed by these 2 Tangier disease patient fibroblasts. And Leanne Davis in Anthony Gaglioni's lab did this quantitation because she's got phenomenal expertise in doing these assays. So to cut a long story short, we found that if you looked at the phenotypes that we find in Niemann-Pick C, which is cholesterol storage, sphingomyelin storage, glycosphingolipid storage and trafficking defects, sphingosine storage, and this lysosomal calcium defect, all of those are present in Tangier. Another really interesting thing was that HM and Ali went on to treat these patient cells with miglustat. And what we found was that these patient cells have lysosomal expansion, just like Niemann-Pick C cells. So we think that they are a secondary lysosomal storage disease because of this secondary involvement of the NPC pathway. But what was really interesting is if you treat with miglustat, shown in these white bars, you can see that the lysosomal volume of these patients was reduced. But when you treat with cyclodextrin, which would normally correct a Niemann-Pick C cell, we're seeing no response. And so this has also raised the possibility that cyclodextrin's benefit in Niemann-Pick C is ABCA1 dependent. And this is the reason that it's not correcting in these Tangier disease fibroblasts. So again, you can see that this sort of interplay between studying how a drug responds in a different scenario and how a patient can inform convergence between 2 rare diseases is a really, really sort of interesting, unexpected axis that we've started to uncover here. So the Tangier study conclusions is that Tangier disease is actually a secondary lysosomal storage disease. We think that cyclodextrin acts through an ABCA1 dependent pathway in NPC patients. And the glycosphingolipid substrate reduction therapies like miglustat and iliglustat may be unanticipated therapies for Tangier patients. And this is important because if you've got an ultra-rare disease like Tangier, people always say there's only about 100 cases reported in the literature, so it's an ultra-orphan disease. There's no incentive for the pharmaceutical industry to innovate therapies for this disorder. But we may have stumbled across miglustat as a potential therapy, and this in turn has uncovered the fact that somehow there is an axis between ABC transporters and the NPC pathway that we're trying to better understand at the moment. Now, this gets even more interesting when you go back to Ali's data that she generated with Doris Hoglinger as part of her PhD. Because what she did was she also ran the screen that I described where she picked up the ER binding partner of NPC1. She also ran another screen where she confined herself to direct protein-protein interactions only on the vacuole of yeast, so in other words, the lysosomal membrane. And she found that NCR1 interacts with a plasma membrane calcium channel that we think has an isoform that is lysosomally targeted. And that protein itself is in a complex with an ABC transporter in yeast. So I think another take-home message from this research that has been another wake-up call to me is that although we study these proteins in isolation and we manipulate them, what we're missing is this wider interactome. And this is suggesting that there is some functional cooperation between ABC transporters and potentially NPC1. So this is leading us to do many more experiments and also try and understand what PMC1 is doing. Is this the protein that's responsible for filling the lysosome with calcium, for example? So I'm just hoping that this gives you a flavour for the diversity of approaches that you can take to try and unpick these very complex pathways. Now I want to finish up not with rare diseases, but with a common disease. And I'm going to just share with you some recent data on why we're getting more interested in Parkinson's disease these days. And again, this is about the observation that was actually made in the clinic initially, that there was a link between the lysosomal storage disease, Gaucher disease, and Parkinson's. And this was missed in GWAS studies. It was careful clinical observation that first made this connection between Parkinson's and a lysosomal disease. And since then, it's become much, much more complicated. And what I want to do is share with you a sort of an emerging role for sphingolipid dysregulation in Parkinson's that goes beyond Gaucher disease. So the Parkinsonism and Gaucher disease connection is that GBA, the glucose-3 biosynthase that removes glucose from ceramide in the lysosome, leads to Gaucher disease. And it was found that the frequency of Parkinsonism was increased in both Gaucher patients, but interestingly in the heterozygous carriers. And mutant GBA alleles are associated with Parkinson's disease in many populations, and I'm sure you're familiar with Ellen Sidoransky's work in this area. And the main sort of controversy, if you like, or the main debate in this field is this a gain of function where this mutant GBA allele complexes with something like alpha-synuclein to form sort of pathological aggregates, or is it actually a loss of enzyme function and there's actually a sphingolipid imbalance? And so what I want to do is just focus on some studies we've been doing looking at this. Now the plot thickened recently when this paper was published. And Josh Schulman at Baylor did a really interesting genetic study where he took a couple of thousand sporadic Parkinson's disease patients, and he asked the question, are any of them heterozygous carriers for a mutation that would cause one of those 70 lysosomal storage diseases? Now, if you look at the 70 odd lysosomal storage diseases that we know of, there are about 60 genes that are responsible for them. And what he found was that over half of the sporadic Parkinson's patients are heterozygous for at least one mutant allele relating to a known lysosomal disease gene. So in other words, is it actually nothing to do with GBA and specifically GBA? Is it actually saying that the risk factor for Parkinson's is lysosomal dysfunction? And the big question is, why is heterozygosity, which normally carriers of lysosomal diseases are normal, why is heterozygosity so overrepresented in sporadic Parkinson's? And some of Josh's, the patients that were studied had up to 4 mutations in different lysosomal disease genes. So this is a really, really fascinating finding. So funded by Parkinson's UK, Mylene Hubecker, who many of you will know, she was a PhD student funded by Parkinson's UK in collaboration with Penny Hallett and Early Isaacson at Harvard, did a couple of really interesting studies where They studied post-mortem material from Parkinson's patients, and they compared Parkinson's patients and normal aging brain samples in comparison. And the focus was on the substantia nigra. So if we look at glucose-3 biosynthase, to start off with GBA, you can see that as you age, and these are the controls are in the open circles, as we age from 70 to 90, our levels of this enzyme go down. But the Parkinson's patients are starting at a lower basal level at 70, and they're going down in parallel with the controls. Now, this GBA activity basically is reflected also by GBA2. So this is the non-lysosomal enzyme. And so this led to the idea that what we're dealing with in the substantia nigra of PD patients and in normal aging is that we're seeing these parallel reductions in at least one lysosomal enzyme and one non-lysosomal enzyme. And what was even more interesting was when Mylene looked at multiple lysosomal enzymes in the substantia nigra from PD patients compared to age-matched controls, she found that multiple lysosomal enzymes declined. So, for example, GBA is reduced, alpha-galactosidase is reduced, beta-galactosidase, neuraminidase, and beta-hexosaminidase. And this means that the potential risk factor in Parkinson's is a decline in lysosomal enzyme function as we age. And she also found that there were parallel changes in glycosphingolipids when you measured these biochemically in the substantia nigra from these PD patients from these brain banks. So, for example, glucosylceramide is elevated, but certain complex gangliosides like GM1A, GD1B, and in fact, gangliosides as a whole are reduced in Parkinson's. And really, very nicely, these changes are also reflected in plasma and serum. So we think these may be biomarkers for measuring these changes in Parkinson's patients. So the question is one of these changes the risk, or is it a combination of all of these things in combination that increase the risk of Parkinson's? And that's something that we're trying to understand at the moment. So to summarize the Parkinson's side of things, we're finding that there's a decline in multiple lysosomal enzymes in normal aging, and this is beyond just GBA. We see a greater decline in the Parkinson's patients, but it happens with normal aging in the general population. It leads to an altered pattern of glycosphingolipid expression in the substantia nigra, in the CSF and in plasma. So this to us is supportive of a loss of function sort of model. There are interesting reductions in complex gangliosides, and what we're trying to understand is why is the dopaminergic system so vulnerable to lysosomal dysfunction? And does any of this provide some new approaches for thinking about treatments for Parkinson's disease? So just to finish up, we've been studying now for many years from rare to ultra rare to much more common diseases with altered sphingolipid metabolism. And I think the spectrum of human diseases which involve altered lysosomal function, altered sphingolipid metabolism, homeostasis, is probably going to expand greatly over the coming years. And we've done this by focusing on both rare and common disorders. And it's giving us insights into sphingolipid function and homeostasis. And it's also hopefully going to lead to new treatment approaches for rare and common diseases. And I think, again, if you're at the start of your career, this is an incredibly rich space to work in because we understand so little, but there are really strong signals that there is a really important part for sphingolipids to play in these disorders. Now, I want to end with some good news. Many of you had signed up and were about to speak at the GRC on sphingolipids. Maurizio and I were the chairs of the 2020 meeting, and Mylene, who I've just shown you, who did all the Parkinson's work, was going to be chairing the GRS, and it was going to be in Il Ciocco, and we were all practically getting on the plane when the whole COVID thing in northern Italy started. And so we cancelled the meeting, which was a really, really difficult thing to do, but with hindsight, I think it was the right thing to do. Well, optimistically, we're now rescheduled. And we have dates that I hope you'll put in your diaries, of the 27th of March to the 1st of April 2022. We're going to be having the first GRS, Gordon Research Seminar, associated with this meeting. And I really urge you all to come and participate at this meeting. I think for the three organisers, it's a bit daunting to be doing all of this twice. And I'd just like to thank the backers of this meeting, including Avanti, for allowing us to carry over the fundraising that we had done to serve the community in 2020. They're allowing us to move a lot of that forward to 2022. So I just want to flag up with you that I very much hope that rather than meeting on Zoom with me in my attic, we will be standing on the terrace at Il Ciocco with a glass of Prosecco in hand and having many, many conversations about sphingolipids in a relaxed and informal environment. So anyway, put those dates in your diary. And Maurizio, if you're on the line, and Mylene, thank you so much for all of your support through this really, really difficult decision-making process relating to the cancellation of the meeting. So I just want to finish by acknowledging a fantastic lab, great set of collaborators, including Sarah Spiegel that we've been working with on the contact site work and the S1P axis in Niemann-Pick-C, and a variety of funders. And we're getting a lot more sort of into Parkinson's these days, but very much looking at the parallels with lysosomal diseases. So thank you very much for your attention, and I hope I haven't gone on too long. I probably have, but apologies for that.